Okay, this is joint work with several people at the University of Texas, including several students, a PhD student. So this is a cartoon, a very simplistic cartoon of how DNA sequences evolve from a common ancestor. So at the root of the tree, you have some sequence of a particular length, and that sequence is evolving down the tree. And it's representing how that particular marker within the genome is evolving just under a substitution model. So if you look at any standard molecular evolution textbook, this is the kind of thing you're going to see. Sequences evolving just with substitutions. And so you have a sequence of a given length at the root, and it evolves, and at the end you have sequences all of exactly the same length at every leaf of the tree. And the problem, of course, is to try to reconstruct the tree from what you observe at the leaves. So that's the problem, and as you see, you get an unrooted tree. You don't get a rooted tree, and that's largely because the models of evolution are time reversible, and so you can't actually locate the root without the inclusion of an outgroup. So you consider yourself accurate if the tree that you get back is correct as an unrooted tree. The problem is that this is not the only thing that happens. Everyone knows that there's also insertions and deletions as well as other events. And so what's going on is you have sequences evolving not just with substitutions, but with what we call indels, insertions and deletions. So this is an example where you start up with this sequence up here. This part is deleted. This T is changed to a C. And so what you normally do is you try to get what's called an alignment. And everyone knows about alignments. I'm just going to define this in the context of evolution. That you're trying to reconstruct exactly what happened in the evolutionary history. So that history of insertions and deletions and substitutions is reflected in the true pairwise alignment, so true positional homology. And if you have a tree and you have sequences at every node and you know how each sequence evolves into the other, then that defines pairwise alignments on the edges and hence by transitivity, the setwise alignment. Okay, so how do you get a tree? Unaligned sequences, you align them, which means you pull them apart to introduce insertions and deletions. And then each column represents positional homology, and on that alignment, you can then get a tree. That's the process. First align, then get a tree. Now, there's a lot of problems with this process, but we wanted to look and see exactly what are the consequences for trying to estimate the alignment. So you can do alignments in many different ways. You can do trees in many different ways. And we're going to fix maximum likelihood as the tree estimation method because it gives the best results for very large data sets. And within maximum likelihood methods, we're going to look at RAxML. RAxML is very good software for large-scale phylogeny estimation. But look at all of these different ways of doing alignments. So there's been a lot of work evaluating how to get a tree once you have an alignment. Here we're saying we're going to fix the tree estimation method, and we're going to say how do you get the alignment and what's the impact of that alignment method. We did a simulation. We published this along with the new method that I'm talking about today in Science last year. The simulation is a simulation with insertions, deletions, and substitutions on large data sets, starting at 100, going up to 1,000 leaf trees. With various different rates of insertions and deletions and substitutions, different indel lengths, so not just all the same length. So this is a pretty highly parameterized and wide-ranging simulation. And then we computed RAxML, so maximum likelihood trees on every alignment. We recorded the tree error, which is the missing branch rate. I'll show you that in a second. And then a standard way of computing alignment error. And this is the way we record tree error. So if you have a true tree and an estimated tree, you can look at each edge. This edge, for example, splits AB from CDE. So does this. So it's a true edge in the estimated tree. Whereas this edge splits DE from ABC, and that doesn't appear here. So this is a false negative. It's an edge that's missing from the estimated tree. So you can talk about the false negative rate or the missing. This is the simulation that we got. So these are 1,000 taxon model trees ranging from easy to hard. And this is just empirically observed easy to hard, okay? So at the left, you get the easiest models. At the right, you get the hardest models. What's changing is the relative rates of insertions and deletions. And so you have more and more what's called homoplazy, more insertions, more deletions, and more substitutions. And in black is what you get if you do maximum likelihood on the true alignment, okay? So if you do maximum likelihood true alignment, your error starts off at 5% missing branch rate and then goes up to 10. On the other hand, if you have to estimate the alignment in these models 7 through 15, having to estimate the alignment really introduces a lot of error, okay? Now this is the alignment error. Okay? Now, this curve is RAXML on the clustol alignment. This clustol is actually the standard thing that systematists use. You can see how badly it's performing. These are other more new alignment methods, but you can see that no matter what you're doing, you're getting a lot of tree error, okay? And it's reflected, alignment error generally reflects in tree error, but not directly, okay? So these models, it doesn't matter how you align. Even though there's alignment error, it doesn't have an impact on the phylogeny. Okay, so what do we observe from this? Current alignment methods really do fail to give sufficiently accurate alignments on large data sets that are evolving with a lot of insertions, deletions, and substitutions. There's so much error that, in fact, most systematists simply discard those markers. They will not use those markers in phylogeny estimation because they don't trust their ability to align them, and so they can't get good trees. And if they want to use those, they're going to manually realign them. And manual realignment is really problematic because there's no rigorous way to do it. It's not really well annotated. You can't repeat it, and it's highly subjective. Okay, so these issues really substantially impact the ability to get deep evolution, because if you're throwing out data because you don't know how to analyze it, you're throwing out information. Okay, so what about doing this at the same time, getting the alignment and tree at the same time? Active research. There are statistical methods that have been developed, like Bayesian methods like Valify, maximum likelihood method like Alifritz. These methods are not scalable. They can analyze data sets with maybe 20 sequences, and even those will take a week or so. They just don't run on bigger data sets. Then there are extensions of maximum parsimony like POI that try to look at minimizing tree length. Those can run on bigger data sets, but they don't give better results than the best two-phase methods. So this is where we came in. We came up with a new method. So that's what I'm going to tell you about now. This new method has a very simple iterative technique where we get an initial tree, we realign on the tree, and then from that new alignment, we maximum likelihood tree, and we repeat the process. So what does it look like? You get a tree, you get a new alignment. This is the novel part. How we do this is our own technique. So you have a new alignment, then you get a new maximum likelihood tree. You look at the ML score under a model that treats gaps as missing data, mind you. It's not actually including insertions and deletions in the model, okay? And if you have a better ML score, you accept it. If you have a worse ML score, you do a different realignment. And just repeat the process and stop after 24 hours or however long you want to run it. So how well does it work? In red, much better. 24-hour analysis run sequentially on a 4 gig machine. Very nice results, just 24-hour analysis. We also looked at a lot of biological data sets where there were curated alignments where we could really have a really good reference tree, and we got similar improvements. So this is the first scalable method for large data sets that can give you trees and alignments at the same time. Now, why does it work? I have to say that this method actually raised a lot of eyebrows in the statistical community because it's not a statistical method that uses a model that includes insertions and deletions. So the fact that it's getting good results is a little bit disturbing to anyone who likes mathematics. So after we published this paper, we tried to figure out why it was working, and we proved that if you actually try to optimize, if you allow the alignment to change and you look for the alignment and its maximum likelihood tree, treating gaps as missing data, if you will try to optimize the ML score that you get in that result, every tree is optimal. So that's a pretty negative result. That says the reason this works has nothing to do with the use of ML, right? Nothing. Because if you really could optimize ML, you get a bad result. And instead, the key is the divide and conquer technique. So this is a method that gets its improvement because it's generating a sequence of alignments, and on that sequence of alignments that it gets in a particular way, it then uses maximum likelihood to pick among those. It turns out that using maximum likelihood to pick among those is not that important. So once we understood that the key was the divide and conquer, we went back and looked at that specific way we realigned. So we have our tree, and we're going to compute a new alignment by the way SATé did this is we took this tree, we broke the taxon data set into subsets, clade subsets, based upon the tree, realigned the clades using MAFT, and then aligned the alignments using muscle. That was what we did. Now, we went and revisited that divide and conquer strategy and changed it a bit. And so we changed the decomposition. It's no longer clade-based, and it's not done exactly this way. And it's a different realignment strategy. It's a new version of SATé, and we get much better results much faster. Here's an example. This is not yet published. We're writing it up. This is the new result. Almost as good as maximum likelihood alignment. So, and in terms of alignment error, better alignment error than everything else. So we're improving trees, we're improving alignment, and runtime is a fraction of the original. So this is very, very nice. Better trees, better alignments, faster. We don't really yet understand why it works, but that's, it's good to have a I don't know why. Summary. So SATé 1 and SATé 2, they actually give more accurate trees, more accurate alignments than the standard two-phase methods. And SATé 2 is faster and more accurate than SATé 1. Why these methods work so well, we don't really understand. It's certainly not really the use of maximum likelihood to choose between alignments and trees. That gives a small advantage, but not a big advantage. If we want to get much better results, we really should develop a statistical co-estimation method that includes cells in the process. But how to do that in a scalable way is really unclear at this time. So acknowledgements. The National Science Foundation supports this. My annual report is due, so I've been working on that. But anyway, this is going into this. This has our downloadable software. This has a biologist-friendly GUI. Any of you who have large-scale data sets that you want to analyze, you can certainly use this. It's very user-friendly. It actually allows amino acid alignments as well. So if you're interested, please check this out. Thank you. Questions?
How does this perform on the protein sequence alignments in terms of speed and as well as accuracy?
Okay, so SATé is not designed to be faster than two-phase methods because each iteration involves running a maximum likelihood analysis on a new alignment. So that's just to start with. In terms of performance, it doesn't improve upon speed over two-phase methods. Two-phase, I mean, first get the alignment, then get the tree. But we do get, the results do match the best two-phase methods. What we haven't yet found are biological data sets of protein data sets which are big enough where this actually has a big impact. The biological, we're matching performance, but they're not big enough where we get the advantage. So the advantage really comes on the big data sets. Excess of 200 sequences before you actually have an advantage doing this. But if you have some data sets, that would be great.
I assume this is based on a simple model of nucleotide evolution for the tree construction?
The simulation is GTR, so general time reversible, with gamma distributed rates, and the estimation for the simulation and for the nucleotide estimation, even on biological, is also RAXML with GTR. So among nucleotide models, it's the most complex model for simulation and the most complex model for estimation.
So would it be possible to extend this to codon models or other more sophisticated
Absolutely.
models?
So this method is very modular in the sense that if you have codon, if you want to do a codon-based estimation and you have a method to do maximum likelihood or any kind of phylogeny estimation, whether it's maximum likelihood or not, under any model, you can just substitute that in for what we're using as RAXML. For that matter, if you have alignment method that you think is better, you can substitute that in. This is a very modular method where you can do substitutions.
Tandy, you said you fixed maximum likelihood. I assume you've looked at maximum parsimony, especially because it's generally faster. Can you give us a glimpse of what the results look like if you change to that, or at least use that as a guiding point to the maximum likelihood tree?
So RAXML starts with the maximum parsimony tree as its initial tree. So what I can tell you is that maximum parsimony analyses of good alignments are a little bit less accurate than maximum likelihood analyses when you get to big enough data sets. So that's one thing. So we have looked at it, but you don't get the same accuracy. And in terms of speed, in fact, a good maximum parsimony analysis takes about as much time as a good maximum likelihood analysis. RAXML and FastTree. FastTree is another ML method. It's not quite as good as RAXML, but now that we've got these methods, you don't really have to switch to MP for tractability issues.
Thank you.
Tandy, I'm more interested in alignments than I am in trees. And I have a lot of protein alignments with thousands of sequences in them, thousands of such alignments, in fact. The trouble is I don't know if they're any good. And what I was wondering is, can you get the speed down by another factor of 10 or 20 if you use neighbor joining or something? I'm willing to give up on the trees. I don't care how bad the tree is as long as the alignment's good.
This iterative process involves getting a new alignment. That's actually pretty computationally intensive. It's not because it's NP hard, but just because the process alignment is computationally and space intensive, right? And then in one iteration, you're doing a phylogeny estimation. You could substitute neighbor joining as your phylogeny estimation, but I think right now FastTree is so fast that you could substitute that and not have the same impact. So you could fine tune this to make it work, and the software is open source, and you can get it. So we can talk about how to do that.
Yeah, I want to know if you took, let's say, 100 proteins, I'm interested in protein alignments, and you align them with the method, and then you add another 100, how much would that affect the first 100? In other words, does the alignment change significantly if you add more data?
So the way that we're doing it, this division into subsets, we align the subsets and then we align the alignments. And when you align the alignments, you don't undo the alignment on the subsets, you just expand them so that they give you an alignment on the full data set. That said, if you're adding taxa but not in the same way, you might want to realign the stuff that you've already aligned. So that question depends upon how you're adding the additional taxa.
All right, a related question. If you threw in random sequences, would it align them or would it refuse to align them?
So that's a question of what you're using on your subset for your alignment method. So if you have an alignment method, so again, we're aligning our subsets using MAFT, and then we're aligning the alignments using muscle or opal. If you want to have an ability to not include sequences that should not be aligned, you just want to substitute such an appropriate alignment method. So this is more of a meta method, and you can substitute things in, get the properties you want.
Oh, all right.
Okay.
Is it easy to
Yes.
change that?
Yes. Make sure I stop when I should stop. Yeah.
The goal of the guiding tree in the multiple alignment is to gather the sequences that are the most similar, so that they are easy to align at the first step. And in a paper, in the Opal paper, they say that it's better to have a guiding tree made by UPGMA rather than by ONG or things like that, because the goal is different from a guiding tree or phylogenetic tree. So I wonder how the fact of having a better phylogenetic tree can improve the alignment.
Okay, if I understand your question, you're talking about the impact of the guide tree on the alignment and the alignment for the purposes of phylogeny estimation. So that's actually a separate thing, and we've been studying that pretty intensively. We have a paper in PSB from a few years ago where we look at that, and it's very interesting. For most methods, but not all, if you give a phylogenetically more accurate guide tree, you will get a more accurate alignment back and hence a more accurate tree afterwards. That said, it's not true for every alignment method. For example, it doesn't seem to be true for muscle. It's very curious, but it is true for MAFT and it is true for PropCons. In this context, we don't realign using the guide tree in the traditional way. So our reason aspect of it, it's not purely a guide tree based realignment where you just go from the bottom up, okay? It's a different kind of realignment step. But guide trees, have a huge impact on some alignment methods, PropCons and MAFT being two of them. A very big impact on prank. Anyone who's using prank should know that it's absolutely essential to give a good guide tree to prank. It doesn't do well with its own guide tree. That's it? Okay, thank you.